Hello! So today I'm starting a series of videos that I'm going to make about each class of macromolecules that's essential to cell biology. So proteins are my favorite, that's why I'm starting with proteins, and this video is just going to cover the structure, what they're made of, not how they work yet, but just the basics, and this stuff is really essential no matter what kind of biology you're in, from basic high school biology all the way to MCAT prep. This stuff is the foundation for a lot of other things that you're going to learn. So let's dive into it. Proteins are polymers. Polymer just means that you have lots of repeating units one after the other, and the repeating unit or building block in proteins is an amino acid. So you should be looking over your functional groups, but just in case, we're going to review. The amino group is a nitrogen, basically, with however many hydrogens it needs to connect with the things it's connecting with. In this case, the amino group is going to be this guy here, the NH2. And when we say acid, we are talking about carboxylic acid. So that's this C double bond OOH group here. This one you'll see represented sometimes as like this. That just means the same thing. So then there will be a hydrogen attached, and this carbon in here we call the alpha carbon. And last but not least, there is an R group or side chain. Either name works. Sometimes they're also called residues, but every R group is going to be different. There are 20 different options for what they can be. They're really diverse in how they are chemically. You have stuff that is hydrophobic or water-hating in this kind of group. You have aromatic amino acids. You have things that can do dipole-dipole forces and hydrogen bonding, like OH groups and stuff. We have our sulfur-containing amino acids. We've got weirdos like proline. And then there are also acidic and basic amino acids. So for example, this proton can get donated away, and that will make this O negative. That often happens with aspartic acid and also with glutamic acid here. The proton will just disappear and you'll see it represented as aspartate or glutamate where it's negative. There's also basic ones, like for example these guys, I drew them already protonated, so they have an extra hydrogen on them, and that will give them a positive charge. So when you're talking about amino acids, you want to associate the basic ones with positive charge and the acidic ones with negative charge. But that's all getting ahead of ourselves because today we're looking at the basics. So how do these things combine into a polymer? Well, they do it in this thing called dehydration synthesis, and dehydration synthesis basically means that we are getting rid of water, or H2O, in order to make a new bond. And that new bond is going to form a group that we see here called the amide or peptide bond. Again, either name works. And you'll have lots and lots of amino acids and lots of amide or peptide bonds. So imagine that we have what's represented here over and over and over again in a long chain. And this long chain is going to have two distinctive ends. One of these ends is going to have the amino group hanging off, and this is called the N-terminus. And one of the groups hanging off at the other end is going to be the carboxyl group, and this one we call the C-terminus. Usually when proteins are synthesized by the ribosome and by tRNAs, it goes from the N-terminus to the C-terminus, kind of the way that DNA always goes 5' to 3'. So how does protein structure work? There are 4 different levels. And in the end, we want to make something big and complicated like the hemoglobin shown up here. The first level of structure is primary structure. Primary structure is just the sequence of amino acids. So something I forgot to mention on the other page is that every amino acid has got a name, it's got a 3-letter code, and it's got a 1-letter code. And all these codes are unique. And since there's 20 amino acids, we're using up almost all the letters. Sometimes they'll make sense, like alanine is A. Sometimes they will not make sense, like tryptophan is W, because we run out of letters. But anyways, you'll see these sequences represented usually with the 3-letter code or the 1-letter code. Like this is the 3-letter codes here, and then we can just make them 1 letter if you want to shorten it, etc. This is what we were talking about with the amide and peptide bonds. That's the way the primary structure is connected. Every structure is going to have its unique set of chemical interactions. For primary structure, it is the peptide bond. Secondary structure gets a little bit more complicated. So when we have a long chain of amino acids, we're going to have a backbone that looks like this. So here's the R group. We don't care about the R group right now. But that's going to be our backbone here with our peptide bonds. And these guys are going to interact with each other via hydrogen bonding to make some pretty interesting shapes. There's 2 common ones. One of them is called an alpha helix, and it's kind of like the DNA helix, except it's only a single polymer instead of double helix of DNA. And the other one is called a beta pleated sheet. So the pleated sheet kind of is like a snake. It twists and turns, and it makes this little shape where each twist and turn is connected. And this, again, is going to be hydrogen bonding between the backbone of the amino acid polymer. Tertiary structure is the overall 3D shape, and here's where things get a little bit complicated because we've got lots of different interactions we can have. So we can have dipole-dipole forces where something that's partially positive, like for example, a carbon is going to interact with something partially negative, for example, a nitrogen or oxygen, and this is going to be interactions in the R group or side chain, not in the backbone. You can also have ion dipoles, so like I talked about, mainly the acidic and basic amino acids can be negative or positive, and those negative or positive can interact with each other in ionic bonds, or they can interact with partial negatives and partial positives, and that we call ion dipole forces. The other thing that can happen is hydrogen bonding between the R groups. So in secondary structure, we had it between the backbone. In tertiary structure, we have it between the R groups or side chains. Another cool thing that can happen is disulfide bridges. This only happens with one specific amino acid called cysteine that has an SH group, and basically you'll have 2 SHs, the H's will come off, and that will leave one open spot on each sulfur to bond with each other. The reason I put a star by this is because I did extensive research in the beginning of my undergrad years with disulfide bridges, so I'm going to be very mad at you if you forget them. Don't forget them. And then we have hydrophobic interactions, and hydrophobic means water-fearing. Amino acids that are just carbon and hydrogen, so basically ones that don't have any dipoles or partial negatives or partial positives, they don't really like to interact with water because they don't have the capability to hydrogen bond or do dipole-dipole forces. So these guys are basically all going to be lumped together saying, hey, let's all go away from water, and they're going to be found on the inside of the protein in something called hydrophobic interactions. It's like in school, if everybody's in a clique and the popular kids all have each other, the losers all have to hang out together, not because they want to get to know each other, but because they're the only ones left. Okay, so quaternary structure is going to be lots of subunits coming together to form a complete thing. So for example, in hemoglobin, there's 4 subunits. You've got 2 alpha subunits and 2 beta subunits. So this is 4 different polypeptides or 4 different polymers of amino acids coming together, and this kind of uses the same interactions that we see in tertiary structure. And this is going to ultimately make things like the hemoglobin shown. So quaternary structure and all these other structures, this is how you carry around your oxygen. This is how you survive. So, conclusion, it's all important. Next time we're going to talk about the function of proteins.